multimodal imaging of a single mouse brain over 5 orders of magnitude of resolution. Sean Foxley A. Vandana Sampathkumar. Vincent Andrade. Scott Trinkle. Abstract. Mammalian neurons operate at length scales spanning 6 orders of magnitude. They project millimeters to centimeters across brain regions, are composed of micrometer-scale-diameter myelinated axons, and ultimately form nanometer-scale synapses. Capturing these anatomical features across that breadth of scale has required imaging samples with multiple independent imaging modalities. Translating between the different modalities, however, requires imaging the same brain with each. Here, we image the same post-mortem mouse brain over 5 orders of spatial resolution using MRI, whole brain micrometer scale synchrotron X-ray tomography, MUST, and large volume automated serial electron microscopy. Using this pipeline, we can track individual myelinated axons previously relegated to axon bundles in diffusion tensor MRI. Or arbitrarily trace neurons and their processes brain-wide and identify individual synapses on them. This pipeline provides both an unprecedented look across a single brain's multi-scaled organization, as well as a vehicle for studying the brain's multi-scale pathologies. Previous article in issue. Next article in issue. Keywords: MRI, synchrotron source X-ray tomography, electron microscopy, whole mouse brain, Connectomics, multi-scale imaging, echoplanar spectroscopic imaging. Abbreviations: MRI, magnetic resonance imaging, DTI, diffusion tensor imaging, automated electron microscopy, MUST, synchrotron-based X-ray tomography. 1. Introduction. The architecture of the mammalian brain is complicated, operating on multiple scales spanning orders of spatial magnitude. Lichtman and Denk, 2011. Neurons in the brain project macroscopic distances, millimeters to centimeters, SR, 1899, while simultaneously producing microscopic connections and plasticity-related changes at the nanoscale. Grutzenler et al., 2002. No current single existing imaging modality spans the 6 orders of length scale, mm to nm, required to map the whole brain while simultaneously resolving individual synapses. Magnetic resonance imaging, MRI, allows in vivo, Vassar et al., 2000, and post-mortem, Dwyer et al., 2007, Miller et al., 2011. Mapping of neuronal tracts spanning macroscopic distances, but cannot easily observe individual neurons, much less identify individual neuronal connections. At the other end of the resolution scale, advances in automated electron microscopy, EM, provide synapse-level resolution, 3 nanometers, Helmstedter, 2013, but remain limited by serious computational challenges. A single voxel of MRI data, 1 mm3, is almost 2 million terabytes of raw EM image data, Bouchard et al., 2016. Thus, EM remains best suited for neuron connectomics, reconstructing connections of individual neurons over limited ranges of hundreds of micrometers. An imaging pipeline capable of bridging these approaches would have to solve the problem of context by providing sufficient information and volume to help register an EM dataset to the relative low-resolution framework of an MRI volume. An ideal intermediate imaging modality would A. Achieve micrometer-scale resolution throughout an entire large volume tissue sample and B. Use a sample preparation compatible with a pipeline including MRI and EM. Synchrotron-based X-ray tomography, MUST, non-destructively provides micrometer-scale resolution over 1 cm 3-hole tissue volumes, the size of a small mammalian brain. Bridging the resolution disparity between MRI and EM imaging techniques, Figure 1. Sample preparation for MUCT uses aldehyde fixed material, compatible with postmortem MRI, Shepard et al., 2009, followed by osmium staining and plastic embedding, compatible with large volume EM, Dyer et al., 2017. Thus, MRI followed by MUCT and EM provides a viable pipeline for multi-scale whole brain imaging. In this work, we demonstrate the feasibility of such a pipeline by imaging a fixed postmortem mouse brain with MRI, preparing the whole brain for subsequent MUCT and EM. Mikula et al., 2012, Mikula and Dank, 2015, reimaging the entire brain with micrometer resolution MUCT, Dyer et al., 2017, Vescovi et al., 2018, and, finally, imaging isolated subvolumes with automated serial EM, Kasthuri et al., 2015. Download high-res image, 1 megabyte. Download full-size image. Figure 1. Imaging modality scale differences capture scale-dependent information. By using an imaging pipeline of MRI, CT, and EM, we can simultaneously resolve brain structures like the white matter at macro-, meso- and microscopic scales in the same brain. We visualized whole brain interconnectivity by inferring white matter tracks from MRI diffusion tensor imaging. The colors denote tracked directions. See inset color sphere. B. We can trace individual axons, white arrows, within the same myelinated tracks over long distances with CT, directly comparing axon paths with tracks produced in MRI. With the addition of EM, we can reinspect the ultrastructure of individually traced axons from B, such as C, the individual wraps of the myelin sheath, yellow arrows. 2. Materials and methods. 2.1. Sample preparation. All procedures performed on animals followed protocols approved by the Institutional Animal Care and Use Committee and were in compliance with the Animal Welfare Act and the NIH Guide for the Care and Use of Laboratory Animals. An 8-week-old female C57BL6 mouse was deeply anesthetized with 60 milligrams per kilogram pentobarbital. The mouse was transcardially perfused with a solution, pH 7.4, of 0.1 m sodium cacodylaate and heparin, 15 units/ml, immediately followed by a solution of 2% paraformaldehyde, 2.5% glutaraldehyde, and 0.1 m sodium cacodylaate, pH 7.4. The brain was carefully removed from the skull and post-fixed in the same fixative for 48 hours at 4 degrees Celsius. The order of serial imaging was MRI, MUCT, and then EM. 2.1.1. MRI. The resected and aldehyde-fixed brain was washed in PBS for 72 hours to remove fixative from the tissue, which can be deleterious to MRI imaging. Diwar BY et al., 2007. Shepard et al., 2009. Just prior to imaging, brains were dried of excess PBS and placed in 10 milliliters Falcon tubes. Tubes were filled with fluorinert, FC 3283, 3M electronics for susceptibility matching and to improve shimming. 2.1.2 MUCT and electron microscopy. After MRI, the whole brain was transferred to 0.1 m cacodylaate buffer. Whole brain staining was performed with multiple rounds of osmium and reduced osmium followed by dehydration and plastic embedding, Mikula et al., 2012, Mikula and Dank, 2015. 2.2. Acquisition protocols. 2.2.1. MRI. Data were acquired at 9.4 Tesla, 20 centimeters internal diameter, horizontal bore, Bruker Biospec Small Animal Mister System, Bruker Biospin, Billerica, Massachusetts. Using a 6 centimeters high-performance gradient insert, maximum gradient strength, 1,000 metric tons per m, Bruker Biospin, and a 35 millimeters internal diameter quadrature volume coil, Rapid Mister International, Columbus, Ohio. The brain was aligned such that the anterior/posterior portion of the olfactory limb of the anterior commissure, AC, was approximately parallel to the magnetic field of the scanner, and the hemispheric midline was parallel to the scanner YZ plane. Using the ParaVision MapHim protocol, third-order shimming was iteratively performed over an ellipse that encompassed the entire brain, but did not extend beyond the boundaries of the Falcon tube/fluorinert interface. B0 maps were produced by recording the voxel-wise frequency of the peak of the resonance, including additional subspectral resolution frequency produced by estimating the maximum peak amplitude of the resonance, described below. This was consistent with previously reported work, Foxley et al., 2015, Foxley et al., 2018, which described a high degree of field homogeneity across samples. DTI was performed using a standard diffusion-weighted 3D spin echo sequence, TR equals 400 milliseconds, T equals 18.5 milliseconds, B value equals 3000 S/M2, delta equals 5 milliseconds, delta equals 11.04 milliseconds, spatial resolution equals 150 isotropic, number of B0s equals 16, number of noncollinear directions equals 30. Receiver bandwidth equals 200 kHz, partial Fourier along first phase encoding direction equals 7/8, duration equals 55 hours 19 minutes 40 seconds. Structural MRI data were acquired using a multi-echo gradient echo sequence. This produces a 4D dataset where data are sampled along time in each spatial voxel to acquire the free induction decay, FID. Sequence parameters were chosen so that the entire voxel-wise FD was sampled to the noise floor, TR equals 1000 milliseconds, T of first echo equals 2.74 milliseconds, echo spacing equals 2.74 milliseconds, number of echoes equals 192, receiver bandwidth equals 75 kHz, flip angle equals 68 degrees, 100 micrometers isotropic resolution. For averages, duration equals 12 hours. 2.2.2 MUCT. The MUCT data were acquired at the 32 ID beam line at the Advanced Photon Source, Argonne National Laboratory. The setup consists of a 1.8 cm period undulator operated at a low deflection parameter value of K equals 0.26. This yields a single quasi-monochromatic peak of energy 25 keV without the losses incurred by use of a crystal monochromator. For a sample 68 m from the undulator, this produces a photon fluence rate of about 1.8 by 107 photons S-1m-2. The X-rays were imaged using a 10 m thick thin film LUAG. Cis scintillator producing visible light images then magnified using a 5x mitotoil long working distance microscope objective onto a 1920 by 1200 pixel CMOS camera, point grade GS3U351S5MC. The effective object space pixel size is 1.17 m2.25 by 1.41 mm2 field of view. The thickness of the thin film scintillator matched the depth of focus of the objective lens, achieving a spatial resolution equivalent to the resolving power of the lens, 1.3 m for a NA of 0.21. Since the camera field of view is substantially smaller than the mouse brain, a mosaic strategy was employed, Vescovi et al., 2018. The sample was mounted on an air-bearing rotary stage, PI Micos UPR 160 Air, with motorized X/Y translation stages located underneath and X/Y piezo stages on top. Typical exposure time for a single projection image at one mosaic grid point and one rotation angle was 30 ms. 3,600 rotation angles were used at each grid point. The sample was translated through a 6x18 tomosaic grid. 2.2.3 Electron microscopy. Regions of interest, ROI, in the brainstem and cerebellum were identified in the MUCT data. The sample block was trimmed to each ROI, and 50 nanometers thick serial sections were collected on aluminium tape attached to a wafer, Kasthuri et al., 2015, and imaged in an Aprio-scanning electron microscope, SEM, Thermo Fisher Scientific. Images were obtained with either 135 nanometers or 3 nanometers in plane resolution with 3 microseconds dwell times. 2.3. Data processing and analysis. 2.3.1. MRI. Data were reconstructed, processed, and analyzed with IDL 8.2, ITT Visual Information Solutions, Boulder, Colorado, MATLAB 2014b, The MathWorks Incorporated, Natick, Massachusetts, 2012, and FSL 5.0.9, FMRB Software Library, FMRB, Oxford, UK. 3D multiple gradient echo data were reconstructed to produce voxel-wise water spectra. Each complex 4D dataset, X times Y times Z times T, was Fourier transformed along time to produce 3 spatial dimensions and 1 spectral dimension, X times Y times Z times upsilon. AT2*. Weighted structural image was constructed from the spectral data, where image contrast was produced by isolating the maximum voxel-wise signal amplitude of the water spectrum, Al-Halik et al., 2002. This was achieved by identifying and storing the voxel-wise maximum peak signal amplitude in a 3D array with the same spatial dimensions as the acquisition. The mean B equals 0s/mm2 dataset from the diffusion acquisition was registered to the structural image via affine transformation using the F-cell linear registration tool, Flirt, Jenkinson, and Smith, 2001. Subsequent diffusion data were identically registered using the resultant transformation matrix. Principal, secondary, and tertiary diffusion directions were estimated using bedposts. Behrens et al., 2003. 2.3.2. MuCT. The acquired subsinograms were registered with subpixel shifting and stitched through pyramid blending into a complete sinogram, Adelson et al., 1984. The center of rotation of the composite sinogram was estimated using an entropy-based approach, Donath et al., 2006. Reconstruction of the entire dataset in whole block mode was performed using the GridRec implementation of direct Fourier reconstruction, Dowd et al., 1999, Tsai et al., 2019, implemented in the TomoPy package, Gersoy et al., 2014, resulting in 11.7 by 11.7 by 17.7 mm3 reconstructed image volume with 1.17 m isotropic voxels. The reconstructed volume was trimmed and stored in 8-bit format. Following reconstruction, CT data were visualized using Neuroglancer, https://github.com/google/Neuroglancer, a high-performance WebGL-based viewer for large, volumetric datasets. Neuroglancer allows for memory-efficient loading of cross-sectional displays across arbitrary planes in the data. This allowed for simple, fine-tuned manual localization of the visible structures in the EM sections within the CT dataset, using large blood vessels and neuronal somas as landmarks. Our CT data can be publicly viewed online through Neuroglancer, http://neuroglancer-demo.appspot.com. Using the tinural link http://tinyurl.com/cxmbjy6b as a significantly shortened, pre-computed source link URL to the general Karturalab. Neuroglancer reconstruction of the data. All manually labeling of somas and myelinated axons was performed using Gnosis, Helmstedter et al., 2011. 3. Results. 3.1. Whole brain imaging with MuCT. Previous work has shown the compatibility of MRI and EM in post-mortem tissue samples, Liu et al., 2011. In order to assess the feasibility of utilizing MuCT as a compatible bridge between the two, we developed a protocol for imaging entire mouse brains, 1 cubic centimeter, at micrometer resolution with MuCT. This included advances in whole mouse brain osmium staining and tissue embedding, Mikula et al., 2012, Mikula and Dank, 2015, extending the field of view of the MuCT system with tomographic imaging, Voscovi et al., 2018, and parallelization of the MuCT reconstruction algorithms using National Lab supercomputers, Argonne National Laboratory, for creating contiguous image stacks from the 10 terabyte-sized raw sinogram data. These developments allowed for both imaging and reconstructing an entire mouse brain at 1 micrometer resolution, Figure 2a. The resulting series of images formed a well-aligned isotropic dataset with sufficient resolution and contrast to capture both individual anatomic regions. Figure 2. The full dataset can be explored in Supplemental Movie 1, as well as underlying brain-wide cytoarchitecture. For example, both neuronal somas, Figure 2c, and long-distance myelinated axon bundles, Figure 2d, could be clearly visualized in the same brain. Data produce sufficient detail and contrast to trace cells, their morphologies, and individual myelinated axons several millimeters, Fig. 2e-G, Supplemental Movie 2. Download high-res image, 1 megabyte. Download full-size image. Figure 2. An entire mouse brain imaged at 1 micrometer resolution with CT. We used high-energy X-rays, 25 keV, for tomographic imaging of a whole, intact mouse brain at 1 μm resolution, collecting 10 terabytes of data in 7 hours. A single downsampled sagittal slice, B, reveals obvious brain regions and myelinated tracts, while the full resolution data contains enough detail and contrast to identify C, individual neuronal somas, nuclei, and nucleoli, as well as D, individual myelinated axons. Since CT data are collected non-destructively over large volumes and with isotropic resolution, we can trace E somas and their neuronal processes and individual myelinated axons, G, over long distances, F. 3.2. Imaging a single sample through the complete pipeline. As described in the methods section, a perfusion-fixed mouse brain was imaged with MRI, prepared for microscopy, and imaged with both MuCT and automatic tape-collecting ultramicrotome, 8UM, serial EM, Kasthuri et al., 2015. Thus, resolutions from each modality spanned 5 orders of magnitude on the same brain, 150 micrometers DTI and 50 micrometers structural MRI data, 1.2 micrometers MuCT data, and 3 nanometers EM data. Typical results, Fig. 3.8d, show similar regions from the same brain imaged with both 50 micrometers resolution T2*, weighted structural MRI and 1.2 micrometers resolution MuCT datasets centered on a coronal slice of brainstem and cerebellum. Using vasculature and neuronal somas as fiducials to guide the subsequent EM imaging, yellow arrows, Fig. 3DE, we found individual synapses, white arrows, Figure 3F, on neuronal somas in the EM data. Thus, we traversed from MRI data of the entire coronal cerebellum/brainstem to the underlying cellular architecture of the spinal vestibular nucleus. With micrometer resolution MuCT data and ultrastructural details of individual synapses with nanometer resolution EM data, all from the same brain. Download high-res image, 2 megabytes. Download full-size image. Figure 3. A multi-scale, multimodal pipeline for imaging the same brain from MRI to EM. The same brain was imaged using diffusion, A-B MRI, 50 micrometers isotropic voxels, C, D, CT, 1.2 /voxel resolution, and EF, large volume serial electron microscopy, EM, 3 nanometers per voxel resolution. VC shows the same FV from the whole brain MRI and CT imaging, corresponding to the red ROI in A. DE show a smaller FV in both the CT and EM data, corresponding to the green ROI in C. Yellow arrows indicate corresponding blood vessels, and a single neuron is labeled purple. Panel F highlights an individual somatic synapse, white arrows on that soma, colored orange. The FV of F is indicated by the blue ROI in E. This pipeline demonstrates the ability to identify corresponding structures in a single brain imaged across 4 orders of magnitude of spatial resolution. 3.3. Comparing results of mu-CT with MRI. We next explored how whole brain mu-CT could indicate underlying cellular correlates of MRI signals. Since both approaches produce isotropic 3D renderings of whole volumes, figures 2A and 4B, this allowed for simple co-registration between the two datasets. For example, Figure 4 shows 3D somas/dendrites traced from mu-CT data from the medial vestibular nucleus. While individually smaller than an MRI voxel, resolving the cytoarchitecture within a voxel suggests a potential source of underlying contrast variability within an MRI dataset. Similarly, the somas in the dentate gyrus, DG, of the hippocampus are easily seen in the mu-CT data, Figure 4C. 10,000 individual cells were counted and traced within the DG over a 50 micrometers slab of data. Again, while the individual somas are much smaller than the resolution of the MRI data, the high density of somas contribute to an anatomically dependent local variation in the MRI signal, rendering it resolvable from the surrounding structures, yellow box. Figure 4. B. Download. Download high-res image, 1 megabyte. Download full-size image. Figure 4. Whole brain CT provides cellular correlates of MRI contrast. B. 3D rendering of structural MRI data used for co-registration with similar CT data, Figure 2A. A. Somas and dendrites of neurons in the medial vestibular nucleus and somas in the C. dentate gyrus were traced from the CT data. While these somas are much smaller than the resolution of the MRI data, they provide possible underlying sources of contrast in the T2* weighted images. Finally, we investigated underlying axonal correlates from mu-CT data with DTI, which is explicitly sensitive to underlying axon distributions. Figure 4A and B show linearly co-registered sagittal cross-sections of the mouse cerebellum and brainstem in both structural MRI and mu-CT, respectively. Overlain on the MRI structural scan are estimates of crossing fibers in each voxel. Specifically, the MRI data supported estimates of 2 fiber populations, Figure 5B1 population traversing the anterior/posterior, A/P, direction and another traversing the left/right, L/R, direction, green and red cylinders, respectively. The yellow boxes in Figure 5A and C indicate the location of 9 MRI voxels, in which 47 individual axons, 27L/R, 20A/P, were traced in the mu-CT data, Figure 5D. The individually traced axons from mu-CT were similarly color-coded for comparison. Figure 5B shows the MRI diffusion. Direction estimates overlain on top of the axons. The populations of axons traced in the mu-CT data clearly follow similar directions to the MRI diffusion direction estimates, while negligible numbers of axons in the mu-CT data traverse directions other than 2 estimated with the DTI analyses. This supports the accuracy of the MRI diffusion model used to estimate directions of underlying fiber populations. Download high-res image, 2 megabytes. Download full-size image. Figure 5. Whole brain CT provides axonal correlates of MRI contrast. A DTI data of the same brain were also acquired. Principal and secondary fiber population estimates are shown overlaid on a sagittal slice of a corresponding structural MRI scan. Green indicates fiber orientation along the rostral/caudal axis. Red indicates left/right, as well as in B, the magnified 9 voxels. C, the mu-CT data from the same brain has been linearly registered to the MRI structural data, and D, individually traced axons from the MCT data are shown with the same color scheme as A and B. Both principal and secondary fiber estimates from DTI data indicate quite similar orientations as the underlying population of individual axons traced from the mu-CT data, as seen by B. The agreement of the DTI direction estimates superimposed on the individual traced individual axons. 4. Discussion. We describe a pipeline using 3 imaging modalities, providing unprecedented views across orders of magnitude of a single mouse brain sample. This will allow for potentially novel understandings and/or interpretations of results within each modality, currently constrained by scale-dependent differences in each individual approach. For example, for axon bundles, MRI data will provide low-resolution, whole-brain estimates of neural tracts. Figure 1a, mu-CT will provide long-range trajectories of individual myelinated axons in those tracts. Figure 1b. Validating and extending the MRI data, and EM would provide reconstructions of individual wrappings of myelin sheaths, figure 1c, or the eventual synaptic targets of those myelinated axons. Because of the contrast mechanisms of each imaging modality, this pipeline works on brains, or any other organ type, of any species. MRI contrast is explicitly sensitive to tissue water, regardless of tissue type. Further, osmium is used as a contrast stain for both mu-CT and EM because it stains membranes of any type of cell in any type of tissue, Pillay et al., 1962. Therefore, in a single brain sample, our pipeline reveals the relative locations of nearly every soma, blood vessel, myelinated axon, and synapse. Other ultra-high-resolution whole-brain imaging approaches often rely on genetic expression of fluorescent proteins to label a subset, often less than 1%, of neurons, limiting these approaches to a small range of tractable animals, i.e. mouse, fly, and worm. Chung and Diceroth, 2013a. Subset labeling, by necessity, precludes detailing how different brain structures, like neuronal somas, vasculature, or myelinated tracts, relate to each other. Finally, whole-brain optical approaches, whether based on expansion. Chen et al., 2015, or optical index matching, Chung and Diceroth, 2013b, often remove cell membranes, making them essentially incompatible with subsequent large-volume electron microscopy. Other clear advantages of using whole-brain mu-CT as an intermediate approach is that datasets are 1. Acquired with isotropic resolution, facilitating unbiased tracing in any direction. 2. Imaged through intact brains with minimal deformation, obviating computationally difficult large-scale alignments. Chung and Diceroth, 2013a, Salfeld et al., 2012, and 3. Compatible with sample preparation for downstream electron microscopy. Such pipelines will likely be useful in studying brain pathology. Pathological alterations in neural function and behavior are well documented, but our understanding of their cellular underpinnings remains fragmentary. The complexity of brain circuitry and the marked heterogeneity of neuronal sizes and shapes across brains could mask even dramatic changes in specific neuronal subsets, making structural analyses difficult. Using mu-CT and EM to inform contrast variability in MRI data over entire brains would significantly narrow the search for pathological structural change and lead to improved specificity in MRI. This would also lead to potential in vivo MRI biomarkers of disease. The next generation of synchrotrons currently under construction, Schmidt et al., 2018. Should allow for even deeper penetration of samples with the possibility of imaging even larger brains in their entirety. Finally, By instantiating our imaging pipeline in a national laboratory, we provide a path for future investigations at a no-cost facility open to researchers worldwide. Ethics Statement. The following ethics statement is in the 2. Materials and Methods subsection 2.1 Sample Preparation. All procedures performed on animals followed protocols approved by the Institutional Animal Care and Use Committee and were in compliance with the Animal Welfare Act and the NIH Guide for the Care and Use of Laboratory Animals. Author's contribution. SF, P.LR, and NK conceived and designed the imaging pipeline. SF acquired and processed MRI data. V.DA designed the CT system used for data collection and contributed in reconstruction of CT data. VS collected all EM data. SD registered MRI, MUCT, and EM data. SF, AS, and CAN trace/segmented MUCT data, and SF registered results to MRI data. SF and NK wrote the final manuscript, and all authors contributed to its revision. Data availability. The data are freely available for viewing, as noted throughout the manuscript. Our CT data can be publicly viewed online through Neuroglancer. http://neuroglancer-demo.appspot.com with http://tinyurl.com/cxmbjy6b as the pre-computed source link. Raw data from any of the 3 modalities will be made available from the corresponding author upon reasonable request. Code availability. All image processing was performed with commonly used freeware tools, noted throughout. Declaration of competing interest. The authors declare no competing or conflicts of interest. Acknowledgements. The authors thank Sean McCullough for help with sample preparation for MUCT and EM. S.F. would like to thank the MRIS and Merck. NK and VS are supported from a Technical Innovation Award from the McKnight Foundation, a brain initiative of the National Institutes of Health, U01MH109100, and National Science Foundation NeuroNext grant. P.LR is partially supported by the National Institutes of Health, R01EB026300. SD is supported by the National Institute of Neurological Disorders and Stroke of the National Institutes of Health, F31NS113571. Partial funding for this work was provided by the NIH S10 Award OD025081-01 and by the previous awards NIH S10 RR021039 and P30CA14599. Additional funding supporting this work was provided by the National Institutes of Health, S10, OD025081, S10, RR021039, and P30, CA14599. This research used resources of the Advanced Photon Source, a U.S. Department of Energy, DOE, Office of Science user facility operated for the DOE Office of Science by Argonne National Laboratory under Contract No. DA AC02-06CH11357. This research also used resources of the Argonne Leadership Computing Facility, which is a DOE Office of Science user facility supported under Contract D AC02-06CH11357. Finally, the authors would like to thank Farron Jane and Jennifer Neely for thoughtful comments and discussions over the course of the preparation of the manuscript. Appendix. Supplementary materials. Download all supplementary files included with this article. What's this? Download. Download Acrobat PDF file, 247 kilobytes. Download. Download video, 30 megabytes. Download. Download video, 5 megabytes. Recommended articles. References. Adelson et al., 1984. E. H. Adelson, C. H. Anderson, BJR, P. J. Burt, J. M. Ogden. Pyramid Methods in Image Processing. RCA ENG, 29, 1984, pages 33 to 41. View in Scopus Google Scholar. Alhalik et al., 2002. H. A. L. Halleck, X. De Fan, M. Zamora, J. N. River, J. E. Mulder, G. S. Karksmar. Spectrally inhomogeneous bold contrast changes detected in rodent tumors with high spectral and spatial resolution MRI. NMR Biomed, 15, 2002, pages 28 to 36. View in Scopus Google Scholar. Basser et al., 2000. P.J. Basser, S. Pijevic, C. Pirpali, J. Duda, Aldrubi, A. J. Mariam. In vivo fiber tractography using DT-MRI data. Magn. Resin. Med. 44, 2000, pages 625 to 632. View in Scopus Google Scholar. Behrens et al., 2003. T. Behrens, M. W. Ulrich, M. Jenkinson, H. Johansenberg, R. G. Nunes, S. Clare, P. M. Matthews, J. M. Brady, S. M. Smith. Characterization and propagation of uncertainty in diffusion-weighted MR imaging. Magn. Resin. Med. 50, 2003, pages 1077 to 1088. View in Scopus Google Scholar. Bouchard et al., 2016. K. Bouchard, J. B. Amon, M. Chun, T. Dean, M. Danker, M. Diveman, D. D. Donofrio, L. M. Frank, N. Kasthuri, C. Koch. High-performance computing in neuroscience for data-driven discovery, integration, and dissemination. Neuron, 92, 2016, pages 628 to 631. View PDF view article view in Scopus Google Scholar. Chin et al., 2015. F. Chin, P. W. Tilburg, E. S. Boyden. Expansion microscopy. Science, 2015, article 126-0088. Google Scholar. Chung and Diceroth, 2013a. K. Chung, K. Diceroth. Clarity for mapping the nervous system. Nat Methods, 10, 2013, page 508. View at Publisher Crossref View in Scopus Google Scholar. Chung and Diceroth, 2013b. K. Chung, K. Diceroth. Clarity for mapping the nervous system. Nat Methods, 10, 2013, pages 508 to 513. View at Publisher Crossref View in Scopus Google Scholar. Donath et al., 2006. T. Donath, F. Beckman, A. Schreier. Automated determination of the center of rotation in tomography data. J. Opt. SOC. AM. AOPT. Image Sci. Vis. 23, 2006, pages 1048 to 1057. View in Scopus Google Scholar. Dowd et al., 1999. B. A. Dowd, G. H. Kimball, R. B. Marr, V. V. Nagarkar, F. V. Tivnis, L. Axe, D. P. Siddons. Developments in synchrotron X-ray computed microtomography at the National Synchrotron Light Source. Proc. SBI 3772, 1999, pages 224 to 236. Google Scholar. Dyer et al., 2017. E. L. Dyer, W. Gray Roncal, J. A. Prasad, H. L. Fernandez, D. Gersoy, V. D. E. Andrade, K. Fetza, X. Shao, J. T. Vogelstein, C. Jacobson, K. P. Cording, N. Kasthuri. Quantifying mesoscale neuroanatomy using X-ray microtomography. eNeuro, 4, 2017. Google Scholar. Dwyer et al., 2007. T. Dwyer, L. David Sogard, G. J. Parker, D. C. Alexander, N. M. Lind, W. F. Baer, A. Hayschmidt, N. Erickson, B. Packenberg, O. B. Paulson. Validation of in vitro probabilistic tractography. Neuroimage 37, 2007, pages 1267 to 1277. View PDF view article view in Scopus Google Scholar. Foxley et al., 2015. S. Foxley, M. Domovich, G. S. Garksmar, N. Schwartz. 3D high spectral and spatial resolution imaging of ex vivo mouse brain. Med. Phys. 42, 2015, pages 1463 to 1472. View at Publisher Crossref View in Scopus Google Scholar. Foxley et al., 2018. S. Foxley, G. S. Garksmar, K. Takahashi. The effects of variations in tissue microstructure from post-mortem rat brain on the asymmetry of the water-proton resonance. Magn. Resin. Med. 2018. Google Scholar. Grutzendler et al., 2002. J. Grutzendler, N. Kasthuri, W. B. Gone. Long-term dendritic spine stability in the adult cortex. Nature, 420, 2002, pages 812 to 816. View in Scopus Google Scholar. Gersoy et al., 2014. D. Gersoy, F. D. E. Carlo, X. H. Shao, C. Jacobson. Tomo Py, a framework for the analysis of synchrotron tomographic data. J. Synchrotron Radiate, 21, 2014, pages 1188 to 1193. View in Scopus Google Scholar. Helmstedter, 2013. M. Helmstedter. Cellular Resolution Connectomics, Challenges of Dense Neural Circuit Reconstruction. Nat Methods, 10, 2013, page 501. View at Publisher Crossref View in Scopus Google Scholar. Helmstedter et al., 2011. M. Helmstedter, K. L. Brigman, W. Denk. High Accuracy Neurite Reconstruction for High Throughput Neuroanatomy. Nat Neurosci, 14, 2011, pages 1081 to 1088. View at Publisher Crossref View in Scopus Google Scholar. Jenkinson and Smith, 2001. M. Jenkinson, S. Smith. A global optimization method for robust affine registration of brain images. Med. Image Anal, 5, 2001, pages 143 to 156. View PDF view article view in Scopus Google Scholar. Kasthuri et al., 2015. N. Kasthuri, K. J. Hayworth, D. R. Berger, R. L. Shalek, J. A. Concello, S. Knowles Barley, D. Lee, A. Vasquez Reina, V. Koenig, T. R. Jones, M. Roberts, J. L. Morgan, J. C. Tapia, H. S. Sung, W. G. Roncal, J. T. Vogelstein, R. Burns, D. L. Sussman, C. Preby, H. Pfister, J. W. Lichtman. Saturated reconstruction of a volume of neocortex. Cell 162, 2015, pages 648 to 661. View PDF view article view in Scopus Google Scholar. Lichtman and Denk, 2011. J. W. Lichtman, W. Denk. The Big and the Small, Challenges of Imaging the Brain Circuits. Science, 334, 2011, pages 618 to 623. Crossref view in Scopus Google Scholar. Liu et al., 2011. C. Liu, W. Li, G. A. Johnson, B. Wu. High field, 9.4T MRI of brain dysmyelination by quantitative mapping of magnetic susceptibility. Neuroimage, 56, 2011, pages 930 to 938. View PDF view article view in Scopus Google Scholar. Mikula et al., 2012. S. Mikula, J. Binding, W. Denk. Staining and embedding the whole mouse brain for electron microscopy. Nat Methods, 9, 2012, pages 1198 to 1201. View at Publisher Cross-Review in Scopus Google Scholar. Mikula and Dank, 2015. S. Mikula, W. Dank. High-resolution whole brain staining for electron microscopic circuit reconstruction. Nat Methods, 12, 2015, pages 541 to 546. View at Publisher Cross-Review in Scopus Google Scholar. Miller et al., 2011. K.L. Miller, C.J. Stagg, G. Duad, S. Fabdi. Diffusion imaging of whole, post-mortem human brains on a clinical MRI scanner.